Good afternoon and welcome. It's great to see you all here in Orlando for the 30th North American Cystic Fibrosis Conference. We hope that this is really an inspiring time for you. Normally this meeting is a time where we bring data, we share data, we form collaborations, and the end result is that we really accelerate our progress towards our ultimate goal, a cure for cystic fibrosis. So we're thrilled that you're here. We're also thrilled that there's a large contingency joining us online. So welcome to them. Would y'all give them a hand for joining us online? And we're glad that you're with us as well. Now, I would first like to invite Cam McLeod, whom most of you know. Is a very remarkable lady. She's a dear friend and chair of the CF Foundation's Board of Trustees, someone who has really had a tremendous impact on this disease. And it's fun for me to watch her in these meetings because although she comes from the hospitality industry, she'll be in the science meetings integrating herself, and she'll come back to me and say, have you thought about this? I'll go, you know, we have. We're going to do that. So, Cam, it's wonderful that you could join us today. Thank you for being here.
Thank you, Preston. It is such an honor to welcome you to the 30th North American Cystic Fibrosis Conference. I'm very thrilled to be here with you today. Whether you work in a care center, carry out research in the lab, or raise the critical funds to advance our mission, you are changing the face of this disease. I'm also thrilled to share that this year's NACFC is our largest ever, with over 4,600 people in attendance from over 40 countries. Amazing. Welcome. I thank each of you for your hard work and dedication to helping all people with cystic fibrosis live long, healthy, and fulfilling lives. I also want to welcome our friends joining us online, especially those of you who have CF. You are the reason that we're here today. Your strength and courage inspire us all. Ours is a team effort, second to none. Fueling our collective effort is an amazing community of CF families, volunteers, and donors. I would like to recognize some of these incredible friends and volunteers from the Southeast and beyond who are joining us in Orlando. Please stand, dear friends, so that we can thank you for your selfless dedication to our mission. I began my journey with the Cystic Fibrosis Foundation over 30 years ago when my son Will was diagnosed at age 2. And I've had the honor of knowing many of you in this room over that time. What a difference we have seen, and what a heritage we have built together. A heritage that we now have the opportunity to share with those of you who are joining us in the fight for the first time. I'd like to take a moment to reflect on just how far we've come together. Wow, isn't that great? And even better, it makes our progress unstoppable. Our collective commitment strengthens the way forward. In our CF family, some have lost a loved one to this disease, as I recently did. Others are currently benefiting from the new drugs treating the basic defect and having a life-changing experience because of the hard work of every one of you in this very large room. And all of us are anticipating treatments in the future that will impact 100% of those affected by CF. For so many families and friends and professionals like you, what begins as a personal commitment to our mission is transformed into a passionate commitment to every person living with CF. What an inspiration to know that all of you, all of us, are one family, my family. I see it when I look out at you, and I have felt it. And all of us dedicated to continuing the journey until the day that CF stands for Cured Forever. Thank you so much for being here, and I wish you all a wonderful conference. And now it's my pleasure, and I'm delighted to welcome back to the stage, to the podium, the dedicated leader of the foundation, someone that all of us admire professionally and personally, my dear friend, our President and CEO, Dr. Preston Campbell.
Well, Cam, thank you, and thank you not only for those words, but for everything that you do. You know, it is inspiring to look out over this great hall and see so many of you here assembled to take on this fight against cystic fibrosis. You're almost, you know, hopefully soon we'll be saying you're 5,000. You're almost 5,000 strong today. That's fantastic. And you, with those who are online and those around the world, are Team CF. And together, you have made the CF story the most amazing story in medicine today. And so why would I say that? I say that because if you go back to when the CF Foundation was started in 1955, there was nothing. There was nothing. There weren't CF doctors, there weren't CF nurses, there weren't CF medicines, there wasn't any CF science. Children with cystic fibrosis were not expected to live to their 5th birthday and ever go to school. Today, thankfully, today things have really changed. The median survival is in the 40s, and over half of our population are now adults. Since 2012, we've added 2 new therapies that treat the basic defect that are truly transformational therapies. Starting with ivacaftor, we started at 4% of the population that benefit, and then it was 8%, and with the approval of Orkambi last year, we moved to 58%, and hopefully in the next 12 months we'll move to 65%. And you'll hear at this meeting that there's a very robust, what we call a second-generation high-throughput screening drug discovery effort that holds tremendous promise, and we think will get us to 95% as we treat those with 1 copy of F508. And the really great thing is that not only will we get to 95%, but we'll significantly raise the level of efficacy for all of those people. Now you may wonder why I have gone through 4%, 8%, 58%, 65%, 95%. Why are we excited about that? Because we know from the approval of ivacaftor in the G551D patient that if we start these transformational therapies early, those people with cystic fibrosis will not have a median survival, how long we expect them to live, in the 40s, or even the 50s, or even the 60s. But we have data that clearly shows that we expect them to live in their 70s and 80s. So you all need to feel good about that, because we've gone from a time when there was no hope to where you have certainly created hope. When we reach 95%, though, and hopefully that'll be in 4 or 5 years or so, as amazing as that will be, we as a community will not be done. We will not stop. We cannot stop until 100% all people with CF have these therapies that address the basic defect. We cannot leave anyone behind, as you'll hear about in our plenary today. And you'll hear it today and throughout the meeting that we already have very significant and promising programs in place to help us reach 100%. But here's the deal. When we reach 100%, you may think we're done at that point. But we're not even done then. Why? Why, when we're at 100% and we think the median survival is going to move significantly forward? The reason is our mission is to cure cystic fibrosis. You may ask, is that possible? We think so. You're going to hear more about that today. But our long-term plan is to combine what is the most amazing story in medicine today, the cystic fibrosis story, with really the most exciting science today, which is gene editing. Bill Skatch and his team already have sort of started this journey, and they've funded over 100 gene editing research grants and formed industry collaborations. This summer, about 120 scientists gathered in Savannah to sort of move this forward and help us realize our dream. You'll hear throughout this meeting that there's already an international effort to apply the new science and technologies to make a one-time cure possible. Our dream is that one day an individual will go into clinic for treatment with cystic fibrosis and leave without cystic fibrosis. Now, we realize that these efforts are going to take a really long time, and because of that, we remain committed to our promise to provide all the therapies that people with CF need. And we continue to invest in new and effective strategies that will develop new novel mechanism of action antibiotics, new anti-inflammatory drugs, new mucolytics, and better nutritional approaches. How's that going? Well, as you saw on the slide, 51 clinical trials are now underway. It's gotten so complicated, our clinical trials group and our communications group had to create a new spot on cff.org, our clinical trials finder that's there for the care centers and patients and families so that they can actually find the trial that best suits them, because we want to make sure that everyone who wants to participate in one of these trials can. Now, I'm relatively new in my role. You all know that. I started basically at this time last year, and I'm often asked, how are things going at the foundation? And my answer is pretty much the same for everybody, and that is that I think things are going great. The reason they're going well, though, has nothing to do with me. It has to do because I have a great, wonderful team. And let me point out a few of the folks so that you'll kind of know who's on the team, on those programs that actually are addressing you. We have other team members that I'm not going to mention today. You're going to hear in a few minutes from Dr. Bill Skatch. Bill and his team are the ones that are creating the science and translating it, including these drugs that treat CFTR and the one-time cure. They have been very successful, and they've created a lot of work for the next step, because what they do is they take their success and they handed it off to Dr. Michael Boyle, who's charged with sort of overseeing the clinical development of all of these programs. Mike and his team, as you can imagine, with 51 clinical trials, are in overdrive continually to make sure that we're able to get this done in a timely way. And then there's the care centers, the core of what we've done for a long time. In 1960s, when the CF Foundation was formed, the first thing that they did is they established the care center network, and with ongoing, constantly improving care, focused on data, sharing approaches, you are the reason, to a large extent, our success today has been so remarkable as far as the improving median survival. And you know that Dr. Bruce Marshall has been leading this effort for a long time in a very remarkable way, and he's recently been joined by Dr. Al Farah, and together they realize that the care center network is really stretched very thin, and they're looking for very creative ways to increase our support with an emphasis on adding more adult physicians, social workers, dietitians, and pharmacists. And as well, give yourself a hand. That's good, right? And they've done a really wonderful, if not beautiful, job of recognizing that mental wellness is a crucial part of an individual's health and well-being. We're providing additional funding for mental health coordinators at centers to better integrate the treatment of depression and anxiety into the existing care plan. We're really proud of that. And beyond that, I'm very excited that they're trying to find ways to use data from the registry to inform care, put it into a dashboard that both patients and families can look at with the care center staff, so that care programs, treatment regimens are co-produced so that it works in the most optimal way for individual CF. And I think that's a wonderful idea, and I want to thank you all for doing that. But it doesn't stop there. We've been busy hiring great people. In addition to Al, we've recently hired Dr. Drusi Borowitz from University of Buffalo to lead our new community partnership program. And it's been established to help us listen to people with CF better and learn from them what we can do to help them live better today's. And at this meeting, Drusi and her team are bringing the voices of individuals into all of the CF Foundation's activities, including a number of exciting live-streamed sessions from this meeting so that everybody can participate. And then I can't forget Mary Dwight. Mary Dwight is fiercely heads up our policy and access program. She and her team are constantly on Capitol Hill and in state offices, making sure that all decisions that are made with the interest of people with CF at hand. And you know, the other neat thing that they've done is they've created, Mary, along with Ann Willis, have created this Compass tool. And I hope that you've seen that. And if you haven't seen it, I urge you to go to the exhibit hall, to the Compass booth, because this tool is really helping people with CF navigate and find access to care and the treatments that they need to be successful with their treatment regimens. And outside of the medical realm, I'm very happy to tell you that we have successfully recruited a wonderful new colleague, Mark Ginsky, who's our Executive Vice President and Chief Operating Officer. He's joined us almost 6 months ago. He has a JD from the University of Pennsylvania. He has an MBA from NYU. He's a team player. He understands drug development, a lot of business activities. He comes to us from Covance, and he's already proved his merit in many, many ways. And so we're thrilled to have Mark join us. You know what I'd like to do, but I don't think I have the time, is to have all of you stand and give yourselves a hand, because you are truly making a difference in the lives of people with cystic fibrosis. You are changing the face of this disease. And it's because of you that we're able to accomplish this. But I have to tell you, as amazing as our story is, we're not done. And we have a number of unwritten chapters ahead of us. And I think that what is going to be true going forward is that this community that continues to grow is going to stay together until we finish this wonderful story. And we are able to kind of look at each other and say, gee, well done. That was really a job well done. So again, I want to thank you all for being here, but let me also introduce some friends by video who are going to inspire you and reinstill that confidence that we're going to finish this together. So look at the video and check it out.
Thank you to the amazing care teams. You're making a difference for me by providing medical and emotional support every step of the way.
Thank you to the researchers and scientists. Because of your dedication, I've been able to work part-time and travel.
Thank you. Because of you, I've been able to move across the country to pursue my Ph.D.
My brother Austin has been able to enjoy his life as a typical teenager.
You inspire my daughter to learn to care for herself as a young adult because she knows she has a bright future.
You're making a difference by giving me new identities I've always dreamed of. I'm not just a CF patient or the girl who's sick. I'm a college graduate, a wife, and a mother.
Say thank you. Thank you.
You inspire me to enjoy each and every day, to love longer, laugh louder, and fight even harder.
I would like you to continue your fight. Even though we've made such progress in the last few years, we would like to reach our ultimate goal of a cure for everyone.
We need you to continue your fight because somebody's got to do it, and I know that I'm not that smart.
We need you to continue this fight because we believe that the answer to this mystery is at your fingertips, and we are depending on you to do the one thing that we as their parents wish more than anything.
Take CF away forever.
Your tireless dedication, your educating us, your efforts to get our CFers into trials, your love and more give us strength of family like nothing else. When the tsunamis of life come at us and we feel like we're drowning, you make us ride the waves. You make the daily dance with fear end in hope.
So again, thank you all for being here. It's now my treat to introduce one of our remarkable leaders, Dr. Bill Skatch, our Senior Vice President of Research Affairs, is going to introduce what I think you're going to enjoy as a great plenary this morning, this afternoon.
Thank you, Preston. Good afternoon and welcome to the first plenary session. As we have just heard, this is an incredible time in CF. It's probably not an incredible time, it's the most incredible time ever in CF. And it's a real honor and pleasure for me to introduce today's plenary speakers and let you hear some of the really exciting science which is taking place behind the scenes. But before we begin, I would like to take a moment to recognize some very important people. Our program planning committee, and especially our co-chairs, dedicate a tremendous amount of time, energy, and effort to ensure this conference reflects the most up-to-date science and care in CF. And they start this task every January, and they work all year long to put together what starts out as a very bare bones outline into what you see all around you now, and all the exciting science and news that you'll hear in the next few days. I would like to ask the members of the planning committee to stand for just a moment while we recognize all their effort and thank them personally. I would also like to thank our sponsoring organizations and exhibitors for their generosity in helping to support this meeting. This year's group of sponsors is the largest ever and reflects our growing diversity and relationships and partnerships and all the programs that have come together to fight this disease as partners. As you pass through the exhibit hall, please take some time to stop, meet some of the sponsors and exhibitors, and give them your thanks. Looking around the room, I realize how lucky we are to have the scientists, the physicians, the caregivers, family members, supporters, community partners, all working together toward a single goal, to cure CF. And it's my pleasure now to remember one of the great leaders who revolutionized our understanding of CF and present the Paul de Saint-Ignace Distinguished Scientific Achievement Award, the highest recognition given for research by the CF Foundation. Dr. Paul de Saint-Ignace was a pioneering scientist and clinician whose legacy will be forever linked to his advancements in CF research. He began his career as a young pediatric pathologist at Columbia Presbyterian Medical Center, working with Dr. Dorothy Anderson. At the time, in the 1940s, virtually nothing was known about CF except the lack of pancreatic enzymes. And Dr. de Saint-Ignace was the first to describe the effects of CF on the lung. Seems kind of remarkable now. And in 1946, he was also the very first person to use inhaled antibiotics to treat children with CF. 2 years later, during a heat wave in 1948, he observed that infants in the CF ward were suffering from heat stroke and dehydration. And that one simple observation over the next 5 years allowed him to show a fundamental defect in people with CF, that they had an elevated salt content in their sweat. We take this for granted now, but at the time, it was revolutionary. The experts thought it was impossible, and not only did it pave the way for the sweat test, which we still use today 60 years later, it was also a critical step in discovering the underlying chloride transport defect and also ultimately discovering the CFTR gene itself. It's humbling to think that when the sweat defect was first observed, the median age of survival was approximately 1 year. We've come an enormous way since then. Dr. de Saint-Ignace also served the CF medical community throughout his 5-decade-long career. He started the first CF center in the US at Columbia. He helped start the CF Foundation. He established the pediatric metabolism branch at the NIH. And through his efforts, he trained many, many physicians, trainees, and scientists who went on to become leading clinicians in the CF field. We remember Dr. de Saint-Ignace today by presenting this award to another outstanding scientist, Dr. Raymond Fursell, who has also spent the better part of 5 decades unraveling the mechanisms of salt movement across epithelial tissues and building the CF community and mentoring the next generation of CF researchers. So let me tell you a little bit about Ray. He was born in Baltimore, and he moved to Pennsylvania at the beginning of high school. And a well-kept secret is that one of his great talents in high school was shearing sheep. Now, you got to keep that in your mind as we go forward. Some of you probably have never seen a sheep being sheared before, but it's a pretty amazing sight. And Ray claims that he was able to shear a sheep in under 3 minutes. I can only say that we're really very, very lucky that he did not pursue this great talent, but instead went the way of other ex-sheep shearers and got his Ph.D. in physiology at the University of Pittsburgh. And during that time at Pittsburgh, really laid a lot of the groundwork for understanding how movement of sodium, chloride, and potassium regulate fluid secretion across epithelia. He moved to the University of Alabama, Birmingham in 1981, where he built the first CF research and development program into one of the premier CF centers in the country. And he also made a very seminal discovery there. He reported in the mid-1980s that cells from CF individuals lacked a particular chloride channel, a cyclic AMP-dependent chloride channel. And he was first to propose that this channel actually might be mutated or missing in CF. And then 4 years later, after the gene was cloned, he was part of the team that actually proved that theory. He returned to Pittsburgh as a professor and chairman of cell biology in 1995, and there he started a second research and development program. He's the only person that has successfully built 2 world-class CF research centers and training centers. Over his career, Ray has published more than 200 scientific papers. He's received numerous awards, and one of his most satisfying experiences was advising the team at Aurora Biosciences, which later became Vertex, where he helped Fred Van Goor and colleagues develop the chloride transport assays that ultimately gave us Orkambi and Kalydeco. Over the past 46 years, Ray has had an immense impact on the lives and careers of hundreds of CF researchers and caregivers, and thousands of people living with this disease. Like de Saint-Ignace, our community would be a very different place without him. For his many contributions, I am proud to present the Paul de Saint-Ignace Award to Dr. Ray Fursell. Please join me in welcoming Ray. Wow.
Well, thank you so much for that. I realized as Bill was talking how old I am. I just wanted to say thank you to all the people who have had the courage to come to our lab and work with us. People like Mike Welsh and Bob Bridges and others that are too numerous to mention, but they've really been responsible for the progress that we made in our own lab. I also want to thank the 3 individuals with cystic fibrosis who have worked in our lab over the years. Sometimes not actually getting to the point where they could finish their work. So those who know me well know that I've had a very difficult summer with my health, but we're on the rebound now. And when I found out from Bill that I was going to receive this award, it was spirit lifting, like nothing else I can ever imagine. So the CF community means a great deal to me, and I cherish it always. And thank you once again.
What an amazing individual. So now it's time to begin the plenary. And I'd like to introduce the speakers for today's session entitled, A Cure for All, Leaving No One Behind. I think we can all agree that one of the most exciting advances in CF has been the development of the CFTR modulators, which restore function of mutant CFTR protein. We know these drugs as Kalydeco and Orkambi, and as you will hear during this meeting, there are new modulators on the horizon that will be even more effective and reach even more people. However, we also know that some people with CF carry mutations that will not benefit from these transformational therapies. And as Preston said just a few moments ago, our goal is to develop a cure for everyone with CF. So we recognize that it's critical to preserve lung function now, not just 10 years from now, while at the same time exploring transformational new breakthrough technologies like gene editing that will, in the future, change the face of CF forever. So this morning, we're going to take the spotlight off of the CFTR modulators, deliberately. Not because we're not excited about them, but to emphasize that new treatments are coming forward very quickly that will benefit everyone with CF, and that in the long run, new treatments will become available that will transform the disease regardless of the mutation. Our first speaker is Dr. Eric Sorscher, professor of pediatrics at Emory University School of Medicine. Eric received his bachelor's degree at Yale and his MD degree at Harvard. So somewhere inside Eric, deep down, I think there's an internal conflict. He was recruited to the University of Alabama by Ray Fursell in 1990. Eric became the UAB CF Center Director in 1995 and served for 21 years before stepping down and moving his laboratory to Emory University earlier this year. Throughout his career, Eric has made many, many important contributions to CF, too numerous to count here. But today he's going to tell us about some very exciting programs that are moving forward to attack CF on all fronts. The second speaker is Dr. Mitch Drumm, who is professor of genetics and vice chairman of research in the Department of Pediatrics at Case Western Reserve. Mitch received his Ph.D. in genetics at the University of Michigan, and he was actually the first graduate student to join Francis Collins' lab. Francis at the time was developing a very novel and exciting technique called chromosome jumping. And shortly after joining the lab, two things happened that really changed CF forever. First, Mitch learned that a childhood friend had a baby that was born with CF. And at almost exactly the same time, he read a scientific paper from Lap Chi Choi in Toronto, which reported a very exciting finding about genetic linkage data of a DNA marker. So these two seemingly random events prompted Mitch to go up to Francis and say, how about we take this chromosome jumping idea and work on CF? And as Mitch tells it, Francis made a phone call to Lap Chi, and the rest was history. Mitch spent the next several years actually building the chromosome jumping library that was used to ultimately clone the gene. And over the last 25 years, he's become a leading world's expert in CF genetics. This morning, he will tell us about some of the very exciting work that's going on in his lab and around the world about gene editing, how it works, and how it could ultimately be used to permanently cure CF once and for all. I'm so very pleased to have Eric and Mitch here today at today's plenary. I think you're in for a real treat. So please join me in welcoming Dr. Eric Sorscher to the stage.
Thank you so much, Bill. It's a pleasure to be here with everyone today and to have an opportunity to present very exciting progress in cystic fibrosis research. You know, the idea of personal therapy is in the future for many diseases, but is a reality now for cystic fibrosis. A drug like ivacaftor is effective for specific patients, such as those with G551D or related forms of cystic fibrosis. Similarly, lumacaftor, together with ivacaftor, is approved specifically for individuals with 2 copies of F508del, so personalized approaches to treatment. Part of the emphasis for today's presentation is that while CFTR modulators continue to advance in transformative and personalized ways, there are also emerging treatments, strategies that result from cutting-edge science and are expected to benefit patients independent of their particular form of the disease. In other words, these approaches to treatment, which might be called symptomatic, can be valuable to every individual with CF, regardless of the underlying mutation, whether you're a patient with G551D, F508del, or something else, it doesn't matter. The strategies are designed to help everyone. So today we'll review 3 main topics. First, we'll spend a few minutes considering ways in which the modulator era is impacting demographics of the CF patient population, what to expect next, and how this information leads to a conclusion that mutation-agnostic strategies, that is, approaches meant to help every patient regardless of the mutation or genotype, they must remain a high priority. Next, we'll review some of the symptomatic treatments that address downstream consequences of CF gene mutation and should be of enormous benefit to CF patients. The initiatives represent a major thrust of clinical testing now, and some will be available soon, so it's important for us to be well informed in this area. It's also important to point out that these symptomatic treatments, they reflect the problems of CF patients, families, and caregivers, the problems that we all face every day. Problems clearing mucus from lungs, with chronic inflammation, with infection. And there's growing understanding of the basis, the scientific basis that underlies these processes, including new ways to intervene. And so we'll survey these strategies at a relatively high level to describe the breadth and the scope of work being done. And then we'll take a fairly sharp turn, and Mitch Drumm will present the remaining part of the plenary with a glimpse into the future. This is a vision in which all patients with CF can have their disease-associated mutation repaired effectively and permanently, and if you'll allow me to be aspirational for a moment, cured of their disease. There are beautiful and elegant molecular technologies, arguably the most exciting in all of biomedicine today, being applied to CF. It's an area our field has now fully embraced. This includes the topical science of gene editing that we've already heard a bit about today, and we'll hear more about that later in the plenary. So let's begin talking a bit about demographics of the CF population in the modulator era. Here's a current breakdown by molecular theratype of cystic fibrosis and groups of CF patients with whom ivacaftor and the combination of lumacaftor and ivacaftor have been approved. Here's a subset of patients that may have access to ivacaftor soon, that may be amenable to treatment with that particular agent. And then in the light blue and the orange are the demographics that reflect target populations for a highly effective series of next-generation modulators rapidly advancing through the clinical testing phase. Shown in dark blue are patients, a projection describing patients without a modulator at this time. The stylized data shown here is for primary airway epithelial cells, and it's emblematic of progress being made in modulator development. If we take the level of molecular repair using ivacaftor as a benchmark, shown here on the left, you can see where we are with lumacaftor and ivacaftor now in individuals who have 2 or 1 copy of F508del. Data from several groups suggests the exciting advent of emerging and highly potent new combination treatments. This includes significant benefit for individuals with just 1 copy of F508del. That is the prospect that upwards of 95% of all people with cystic fibrosis may achieve a level of repair at or above that seen among CF patients receiving ivacaftor. We're not there yet, but intense efforts are being levied in order to assure success. Beyond the demographics and the epidemiology and beyond the science is the urgency of what really amounts to a historic effort. It's the human effort being extended worldwide to ensure we reach our goal. Many of the groups pursuing the modulators are summarized on this slide. You can see there are a large number of compounds rapidly advancing towards the clinical testing and through clinical testing phase. We'll hear more about premature stop variants or X mutations later in this session, but it's important to note that patients with this form of the disease are representative of individuals for whom an effective modulator compound is not yet available. Clinical data from the most recent Adalorin study is expected next year, and just as with other new CF treatments, there's growing enthusiasm and momentum regarding ways to repair X or premature stop variants. So part of the effort directed towards getting a treatment to every CF patient includes programs directed specifically to premature truncations, and these programs include the one at PTC, a program at the University of Alabama Birmingham in Southern Research, the expanding CFFT laboratories in Massachusetts, as well as other pharmaceutical groups and academic laboratories, all of which are aggressively moving forward in this area, again to make sure that something's available for everyone with CF. Just as an example of the sort of work being done for premature stop mutations, the CFFT laboratory provided data shown here from a screen of 85,000 different compounds to find agents that would have the potential to help. The CFTR, the protein responsible for CF, normally shows up as a band around here on a standard protein gel. But for X mutations such as Y122X, you won't see any of the full-length CFTR shown by the oval that looks empty. And that's the problem. We do have drugs that partially overcome this problem, such as G418 or gentamicin with or without other agents in combination. And you can see the levels of repaired protein that are possible. You can see that it makes a difference, and that in model systems they clearly make things better. Here are some experiments over on the right that show that the same drugs can be used to activate CFTR function. So if this is the baseline function for 3 different X mutations that we see in the clinic, the upward deflection here is indicative of improvement. And so these drugs mediate improvement. Now, the CFFT laboratory has been going about the business of discovering new compounds that very effectively repair X mutations. So here's the signal from a new drug that basically blows the gel out. So while there's a lot of work that remains to be done, this is a remarkable signal. And nothing like this has been seen by the community, the research community previously. The point is that our research community has set the goal so that no patient will be left out. And it's vitally important that every patient, regardless of where they are in the demographics or the pie chart, regardless of the prevalence of their mutation, that every patient needs, deserves, and ultimately will have an effective modulator treatment. So how do we achieve this? It's not easy. The demographics of CF indicate over 1,700 variants associated with clinical disease. So the complexity and challenge of theratyping, that is finding drugs that work for specific mutations, is significant. But several features of the challenge work to our advantage. First, there's a laser focus on this objective. And in international consortia, investigators throughout the world who have set this goal as a mission priority, and rapid progress is being made. In addition, we're finding specific modulators that work in some unexpected settings, and for some patients we think of as needing a certain type of modulator, the same individuals may benefit from new compounds in unexpected ways. For example, we didn't know whether ivacaftor would work for other forms of CF before G551D when it was identified, and now it's clear the compound has beneficial activity in many, sometimes unanticipated forms of CF. In addition, new endpoints are being pursued that will help us predict who responds to emerging CFTR modulators so that we can move these new drugs towards approved indications and third-party reimbursement. There is great urgency, and progress is being made every day so that a greater number of patients will benefit from these types of treatments. As more patients benefit from effective therapy with CFTR modulators, and as more CF genotypes are treated with better modulators at younger ages, the health of the CF population will shift, and it's likely to be dramatic. It's possible to model this epidemiologic scenario. It requires assumptions such as key patient needs, risks or barriers to achieving a particular endpoint, and alternative strategies to consider. But there's no question that the patient population will change and patient needs will change, and in fact, this is already occurring. Here's one model projection showing patients receiving effective modulator treatment. The point is that during the next several years, increasing numbers of CF patients are expected to have received highly effective modulator treatment, shown here in blue and green, so that the proportion of individuals without an effective modulator will diminish. That's shown in the orange. But it's also important to point out that many of the CF patients who receive effective modulators will be individuals with established and pre-existing lung disease. This is the case today for many whose condition has been improved or improved with ivacaftor or lumacaftor. These patients are expected to live longer and better lives, but may still benefit from symptomatic therapies for their existing CF lung disease. These sorts of demographic projections lead to a conclusion that new and better respiratory treatments will continue to be needed over the next several years, and for several reasons. First, until we have the modulator combinations for all CF patients and show they're safe and durable, we're not there yet. We need better lung-directed treatments for many patients with CF, and we need them as soon as possible. Second, there are patients who don't yet have a highly effective modulator treatment. For example, patients with a premature truncation or a splice site defect or a specific problem with the CFTR ion pore. Third, even among CF patients whose lung disease is markedly improved or stabilized by effective modulators, many in the coming decade will still experience respiratory disease that can be helped by symptomatic lung treatments. So if modulator therapies are intended to address the fundamental CF defect, symptomatic therapies are designed to overcome downstream sequelae. And we've discussed the changing demographics for CF patients and the evolving and vital role of modulators. We'll hear more about modulator treatments tomorrow in Plenary 2 from Patrick Flume. What I'm going to discuss and focus on now is some exciting data regarding downstream or symptomatic treatments. So just what are these treatments? Here are the interventions we've been discussing so far, the growing number of robust modulator combinations, and the exciting topic of CF gene repair and editing, which will be discussed by Dr. Drumm shortly. But a substantial part of what goes wrong in CF, in the lungs, the pancreas, the liver, the intestine, can be quite independent of a specific mutation and involves inflammation, clearing mucus, and infection. For many patients, these aspects of the disease are as vital as the advent of highly effective modulators. The science that underlies these processes is advancing rapidly, and it's science that drives new therapies, the formation of mucus, the reasons for its obstructive properties, and the causes of infection and inflammation not yet fully understood. But recent work in the CF pig model, as well as in the CF ferret and rat and mouse and rabbit, are allowing us to ask questions we could never ask before, questions about how mucus is formed, where inflammation begins, why infections get started in the first place. So let's consider now mucus obstruction and the ways that we might overcome this issue. Now, for the first time, we can comparatively study the CF mucus defect in an intact airway in the laboratory. For example, this experiment shows mucus production and clearance from the surface of a non-CF pig trachea. And then the same sort of experiment from a CF pig. The tracheas are submerged in saline, and fluorescent nanospheres are used to monitor the mucus strands. Once again, here's the non-CF and the CF. You can see the dramatic situation in the disease. You can see that in CF, the mucus strands fail to detach from the tissue surface. They can't be cleared. And in this way, they contribute to defects of mucociliary transport. These strands are formed in submucosal gland structures. They travel down the ducts, shown by an SMG here. They emerge on the surface of the airways, above the surface epithelium. And they're made up of constituent mucins that can be fluorescently labeled or profiled with specific lectins. So here you see two types of mucins, MUC5A shown in green and MUC5B in red. These are two of the major mucins that comprise human mucus and CF mucus as well. If you want to look at this in 3 dimensions, it looks like this. You have to agree, this is far from the way we're used to looking at mucus in a sputum cup. And since the type of mucin can influence the propensity for infection, inflammation, or obstruction, from basic science studies like these, we're learning much more about the fundamental problem in CF lung disease and what can be done about it. As mucus strands form, they affix themselves to the airway surface. If this is the surface of the airway, the mucus is shown here plugging a submucosal gland duct, extruding onto the surface of the epithelium and plastering itself to the surface, to the epithelial surface of the airway. And drugs such as DNase and hypertonic saline are already available that work by breaking up or mobilizing mucus, the mucus lining the airways, so that it can be lifted off the airways and cleared. And this slide shows examples of new agents being developed to promote mucus clearance in CF lung disease. There are a number of strategies for accomplishing this goal. Drugs such as hydrators can be added externally by aerosol. They can be used for this purpose, to hydrate the mucus, to lift it off the airway surface. Mucolytics can be used to break up the mucus so that it can be more effectively cleared. Blockers of the epithelial sodium channel, which normally sits along the surface of the airways and reabsorbs sodium so that chloride follows and water follows and decreases the airway surface liquid level, desiccates the fluid, and is partly responsible for mucus plastering to the surface. If this can be blocked, then the mucus itself will lift off the airways better. All of these have the potential to be interventional strategies of great significance, and all are advancing in all of these areas through the clinical testing phase or making progress preclinically. All are exciting and all have the potential to be used in combination with each other. They're shown here. Now, treating pulmonary infection has been a mainstay, of course, of CF therapy. A reason for better outlook among most patients with the disease has been availability of robust antimicrobials for treating respiratory Pseudomonas or other bacteria. There are numerous groups pursuing highly active drugs for this purpose, either through inhaled antimicrobials or systemic drugs, and there are specialized programs in place to target specific pathogens in CF lung disease, the pathogens that are so important to all of us. For example, this slide shows a limited study of patients with G551D CF given ivacaftor, and on the log scale, you can appreciate the way Pseudomonas counts fall during the first year of therapy, but actually may rebound afterwards. This result again emphasizes that a robust pipeline to address infection and inflammation will be needed in the post-modulator era. I wanted to give one example among many of new treatment strategies which have grown out of the Foundation's research development program. This approach is based on the strong requirement for iron when Pseudomonas grows and forms biofilms. To a bacteria like Pseudomonas, gallium looks a lot like iron, but gallium is actually damaging to the bacteria, so Pseudomonas dies because it attempts to use gallium instead of the iron it requires. Gallium represents a potential low-hanging fruit then for treating CF respiratory infection. It's already approved by FDA for a different clinical indication unrelated to its antimicrobial properties. In this experiment, a single injection of gallium reduced mortality in a Pseudomonas pneumonia model in mice, and gallium was also found to be safe and effective in a small pilot study which is now being expanded to phase 2 testing. This is again an example of the strengths of CF basic research, the Foundation's investment in individual research laboratories, the research development program, the consortia, the therapeutic development network, all mutually reinforcing and making translational projects like this one possible through the dedication of scientists worldwide. Just as mucociliary clearance and antimicrobial pipelines are extensive, so are the prodigious efforts to treat CF lung inflammation. A simplified view of the process is shown on the next slide. We wanted to really make this very simple so that everyone could get all the details. Did I mention this was simple? Well, it is complex. But an important principle has been to target the best established culprits, such as the neutrophils or white blood cells that infiltrate CF airways, or elastase released by these neutrophils that can cause damage to lung tissues. There's nothing subtle about the sheets of polys in CF airways. Another point to make is that the field is using multiple approaches to hit multiple pro-inflammatory targets. So a few salient examples. The Celtaxis program looks at a means of blocking sheets of white blood cells that enter CF mucus. A project from Corbus uses a potent inhibitor of inflammation and is investigating better ways to augment resolution of inflammatory disease in CF lungs. Laurent is working towards correcting lipid imbalance, believed to contribute to CF respiratory decline. Gilead has a MMP9 antibody inhibitor that will address lung inflammation and inflammatory disease, and Polyphore has an emerging technology using a peptide to block neutrophil elastase. All of these are moving forward in the clinic. Development of new and effective anti-inflammatory therapies in CF may require a somewhat specialized approach. For example, it's expected that a clinical result may require a trial that lasts several months, and better endpoints may be needed to predict clinical benefit. In addition, care needs to be taken in studies like these not to block the immune response to such an extent that bacteria gain an upper hand. So in summary, the powerful CF resources and research tools shown here on the left, and our commitment as a community and a family to working towards a common and unifying goal have led to unparalleled programs that will improve treatment and outcomes for CF respiratory disease. Throughout this meeting, we'll also hear about efforts to address the critically important non-respiratory manifestations of CF and how the same sophisticated capabilities being used to better CF lung disease can be applied to other organ systems, and these advances will be discussed during symposia and workshops over the next several days. But now, as promised, it's time for a glimpse into the future, a future in which we'll have even more to offer as we begin to effectively target the primary CF gene defect in the DNA sequence itself. I've introduced some remarkable efforts underway to advance new treatments designed to benefit all CF patients regardless of genotype. These are progressing clinically. Getting treatments to every patient is a lofty but essential goal. We've discussed the excitement engendered by the modulators. We've also talked about specific therapies that are intended to treat downstream sequelae, infection, obstruction, and inflammation. These are all expected to reach greater and greater numbers of individuals, all towards the goal of achieving treatment for everyone with cystic fibrosis. One of the challenges we face is that our success as a community means there are many clinical studies that need to be done in order to evaluate new ideas and their role in treatment, and completing these trials is an important challenge that we'll hear about more in the plenary session tomorrow. So now we'll go from the topic of modulators and symptomatic treatments to a very molecular vantage point. Directly repairing the DNA changes that cause cystic fibrosis, this is the ultimate approach to treating every patient with CF. It's vitally important to understand these efforts and how they're envisioned to provide a global therapeutic strategy, and in addition, how they'll impact our objective of seeking a cure for all. Progress is being made towards repair of CFTR mutations themselves, which my colleague and friend Mitch Drumm will talk about next. Mitch is from Cleveland. Mitch is from Cleveland, so I wanted to apologize in advance for any insufferable baseball banter he might introduce into his part of the plenary. I have no particular ties to Chicago or the Cubs. That's my only disclosure. But I wanted to make sure the overall plenary was balanced. Thank you. Mitch Drumm will now come up.
Thank you very much, Eric. Yeah, I was going to throw in a plug for the Indians, but I thought that would just be inappropriate. So, okay, sorry here. So as Dr. Sorscher so nicely has summarized, the goal of the CF Foundation and the research they support is to discover and apply therapies that will improve the lives of all CF patients. You've heard that theme again and again today, and that really is where we're trying to go. And you just heard about the incredible progress that is being made with drugs that target the symptoms of CF, like the anti-inflammatory drugs, the drugs to improve mucociliary clearance, and the antimicrobials. So those are relevant to all CF patients, not just those with a particular genotype. And tomorrow you're going to hear much more about the recent advances with the CFTR modulators, those correctors and potentiators that are being applied to more and more patients every day. So the efficacy of these drugs, though, has been so great, it has firmly established the validity of CFTR as a target, right? So it can help the disease. So now, as Dr. Sorscher alluded to, I'm going to tell you about some new technologies that we think may be the future of CF therapy for all patients. And those work through manipulation of the CFTR gene. And you won't be surprised, I think, to learn that once again the CF Foundation is really leading the way on this, that they've got the foot on the gas pedal, and in keeping the roadway analogy going, there's no stopping any time. We are going full steam ahead. So I'm going to tell you about some new programs, new technologies that just a few years ago were totally inconceivable that we might be able to apply these to CF. But now they're a reality, and we have substantial evidence that we can manipulate the CFTR gene for therapy. All right, so how do we attain a therapy targeting CFTR when not all patients have the same CFTR defect? And you've heard about the different classes of mutations over the years, and I've diagrammed them here to remind you. And so we have the class 1 mutations they have mutations that don't allow any CFTR to be made. We have the class 2, 3, and 4 mutations that produce CFTR, but it's not functional for various reasons. And then you have class 5 that make maybe a little bit of normal CFTR, but not enough to prevent disease symptoms from happening. And you just heard about many of the compounds that are being tested for their ability to improve CFTR function of the class 2, 3, and 4 mutations. And you'll hear more about that by Patrick Flume tomorrow. But as I mentioned, the success of these compounds is just an incredible precedent. It's shown us that we can stop the progression of CF and even improve the health of CF patients by bringing back CFTR activity to the cells in their bodies. That's where we can go. All right. But what about those class 1 and class 5 mutations? What do we do with those? Those are the 5% of patients that have both copies of their gene have these stop mutations, as we call them. There's no CFTR there, so the modulators aren't going to work, right? So, and I'll show you a few examples of them here. Things like G542X, W1282X, and a splicing mutation that I call the mouthful mutation, the 3849 plus 10 KB C to T. that most of you probably haven't memorized. So let's think through this process for a moment here. So if there's no CFTR protein produced, we need to find a way to make the cells produce functional CFTR. And so we have to start at the beginning, working our way back to the basic defect in the DNA, the mutations in the CFTR gene that ultimately cause CF. So obviously being able to treat the mutant protein with CFTR modulators is an incredible precedent-setting leap in that right direction. But there are a few steps, even before CFTR protein is made, that we can, that provide us additional targets to go after. And they're not only applicable to CF, that this is setting the stage for any genetic disease. So what we learn in CF, we can apply to many, many other genetic diseases. And I think that's something that we oftentimes overlook, that the rest of the world is taking advantage of what CF and the CF Foundation have provided. So anyway, it all begins with your DNA, and that's one of the key components of your chromosomes. So I'm going to take you, well, if you're my age, I'm going to take you back to college, but if you're, say, under 50, I'm going to take you back to 10th grade to your biology class and into a cell and remind you how a gene works. That the DNA in the nucleus of the cell has segments that we call genes, and those are copied by the cell into RNA molecules. And that RNA then gets processed while it's in the nucleus, and it gets into what's called a messenger RNA or an mRNA. And that is then exported out of the nucleus into the cytoplasm, where it's translated by the ribosome and into a protein, and that protein folds up into a particular conformation and can have its function. Well, that function for the CFTR protein, as you well know, is to allow anions like chloride and bicarbonate to move back and forth across the cell membrane. And in the lungs, that property allows the lungs to maintain the fluid balance that's needed to clear bacteria and other debris that gets in there and allow mucociliary clearance to operate as it should and keep the lungs healthy. Right? But when you have a mutation in the gene, it disrupts the process in a number of different ways. And those that cause a defective CFTR protein, like delta F508 and G551D, those actually react or affect the protein, effectively plugging it as if it had a cork in it. Right? Well, we have these new drugs, these modulators, and lots more on the way that, again, you'll hear about tomorrow, that can act on the mutant protein and allow it to open up so those anions can get back and forth across the cell membrane and restore function to the airways. But as we've mentioned, many of the mutations result in no CFTR protein being made whatsoever. And so these you may know by a variety of names. We talked about class 1 mutations, but also they're called nonsense mutations, frame shift mutations, splice mutations, stop mutations, or even X mutations. And these mutations prematurely stop the translation process from happening normally. And CFTR is not produced by the cell then. And so for those, we need a strategy that will fix the steps before the protein's made. And that's what we're here to talk about. Right? So move back before the protein. What are your options? Well, you've got DNA and you've got RNA, and as far as we know, those are the only things that precede the protein production. So let's start with those two. Starting with the DNA, why would we think that we can fix the DNA? Now, our cells are pretty darn good at protecting us from DNA damage. So there are lots of barriers that prevent us from changing the DNA. But there is new technologies out there that were discovered just a few years ago that make us think that we can overcome those barriers. These technologies we refer to as gene or genome editing, and they comprise a way to alter the DNA sequences, making very specific and precise changes in the chromosomes in ways we just didn't think were possible. All right. Now, let me remind you also that the CFTR gene is one gene out of about 20,000 or so across our chromosomes in our genome, and our genome is comprised of about 3 billion subunits that we call the nucleotides, the A's, the C's, the T's, and G's, that give the cells all the information they need to make us who we are and what we are. And some parts of our genome are fairly forgiving, that some places you can change the DNA sequence a bit and it won't have much of an effect, but other places, like the CFTR gene, are actually very unforgiving. And changing as few as one base pair out of that 3 billion can cause a very devastating disease, as you all know from your CF experiences. And I'm showing you here just a small segment of the CFTR gene, the DNA sequence from a region called exon 12. And this particular example is showing the CF-causing mutation G542X. And that happens when a single G is mutated to a T. In this case, it creates one of these stop mutations that prevents CFTR from being produced by the cell. And by changing just this one base, a stop signal is created and the entire gene is compromised. Okay. All right, so how do we go about, I've shown you what's happened when it's broken, but how do we repair that process? And the process of gene editing relies on making a break in the DNA at or near the site of the mutation, and then tricking the cell into replacing the mutation with normal sequences when the cell repairs the break that we caused in the DNA. And the precision of the process to target a specific mutation site is provided by the very complexity of our genomes. So each gene has a unique arrangement of those nucleotides, the A, Cs, Ts, and Gs, so that the regions of our chromosomes are effectively barcoded. And then with that barcoding, we can then very easily select what region or what sequence we want to target. And until the current editing strategies were identified, the ability to do that was just inconceivable. DNA breaks were low occurrence and essentially random. All right, so how this works is that we have to get a couple of different components into the nucleus of the cell. And once we get them in there, the process is to use a targeting system. There are several of these. I'm going to talk a little bit more about the CRISPR-Cas9 system. But that system then locates the position on the chromosome where the mutation's located, and that's what we're going to try to repair. And when that sequence, when that site is found, the Cas9 will break the DNA. And the targeting part of this is possible because of the inherent barcodes that I mentioned. If we add a template DNA that has the normal sequence that we want to incorporate, then the sequence is introduced during the repair process. I'm showing you here in magenta is the T mutation that causes G542X, and with the template that has the normal G at that position that we want to incorporate into the chromosome. And to try to give you a little better idea of how this works, we put together a video that I think will help you conceptualize how the process can occur. I just have to, I'm sorry, I'm missing the, thank you.
In cystic fibrosis, thick, sticky mucus presses down on cilia inside the lungs, preventing the cilia from beating and clearing the mucus. The cause of this problem is an abnormal CFTR protein created from genetic mutations inside the cell nucleus. Our goal is to replace and repair the abnormal DNA sequence to create a functional protein and restore normal mucus. One way to do this is to use gene editing technologies that cut out and replace defective parts of DNA, such as CRISPR-Cas9. The CRISPR-Cas9 repair system has 3 components: a guide RNA that seeks out and binds to a specific location in the DNA, the Cas9 nuclease enzyme, a type of molecular scissors that cuts the genetic sequence at the precise binding site, and a repair template containing the correct genetic sequence for the gene. Once these 3 components are introduced into the cell, the guide RNA binds to and activates the Cas9 nuclease. Now the Cas9 nuclease, the molecular scissors, can search the cell's DNA for potential targets. At a potential target site, like the one shown here, Cas9 unwinds a section of DNA to see if the guide RNA sequence matches. If the DNA sequence matches the guide RNA, the Cas9 nuclease cuts the DNA to create a break in the strands. To replace the mutation with a normal sequence, a repair process is used that precisely edits the DNA. For this editing process to work, a short piece of DNA template that contains the desired normal gene sequence is introduced into the cell. The cell uses this fragment as a template and copies the new sequence as it repairs the broken DNA. Once the first strand has been repaired, the template falls away, and the other strand of DNA uses the repaired strand to fill in the remaining gap, leaving both strands with the corrected gene sequence. The cell then uses the corrected DNA strand to produce a functional CFTR protein that allows the proper flow of salt and fluids through the cell membrane. Mucus returns to normal within the lung, allowing the cilia to beat freely and clear the lungs of germs and irritants.
So hopefully that gives you some idea how this process works. And now the next question, where do we start? You're aware there's a lot of CF-causing mutations. You heard the number. There's over 1,700 CF mutations, and they're spread out across the 190,000 some base pairs that make up the CFTR gene. But we've told you we're going to focus on mutations that are carried by that 5% of patients for whom no CFTR is made. And again, just to put this in perspective, looking at the real numbers that are out there. So 5% carry both copies that have that mutation. But if you look at the CF population who carry even one copy of that, it's about 12%. And that translates into thousands of patients. So just looking at the top 10 mutations in these groups, you can see the numbers, over 4,000 patients carrying one of these types of mutations. So this is clearly a pertinent type of mutation we have to go after. But many of these mutations are found in just single individuals, or maybe a very few number of patients. But looking at them together as a group, they account for, as I mentioned, thousands. But for the patients who carry these mutations, these are clearly the most important mutations that exist. And we need something for them. So these mutations unquestionably a significant problem for us, and we hope to implement these gene editing strategies for them. But to do that, we need to also think about and understand the challenges and the obstacles that we're going to face to do that. And there are two generic ones that we need to think about. One is changing the DNA where and only where we want to, what we would call the precision and the specificity of it. And the other is actually getting the components into the cells that we want to. And that we refer to as delivery. Well, with regards to the specificity and precision, we can get CFTR and only CFTR targeted and make the exact changes we want to. And in the lab, that's not really a big problem. But in vivo, that's another issue. Now remember, the CFTR gene is about 190,000 nucleotides, and of the 3 billion that make up our chromosome. So on one hand, it's quite large, but on the other hand, in the grand scheme of the whole genome, it's actually a small component. So to tackle this, the CF Foundation has done again what they always do, is they go out and they look for the people who know how to do things. So they have started working with various companies. So they've contracted with Editas Medicine. These are the folks who are really the experts in the field of gene editing, and they have been engaged for this process. Others, CRISPR Therapeutics, working with Vertex Pharmaceuticals to try to tackle some of the same questions. So bringing together the folks who know CF and CFTR with those who know how to do these editing strategies, that's the approach. All right, another challenge that we face that I think maybe have been obvious to you is the variation in CFTR itself, the distribution of these mutations across the gene. Now, we can very precisely target DNA breaks now, But that process only works over about a 50 base pair stretch. So remember, you got 190,000 base pairs, and we can target about 50 base pairs at a time. So that provides a bit of a conundrum for us. We can't do them all. So I'm going to try to further the travel analogies here and try to put this in perspective. So think about this. If we make CFTR and stretch it across the U.S., from LA to Boston, that's about a 3,000-mile trip. And those of you who travel the U.S. roads, you know that we like to decorate our road mutations with these beautiful orange barrels. And on average, you'd find one of those barrels about every mile or so. But our CRISPR repair crews can only work on stretches of less than a mile at a time. So we're going to have to be strategic about which of these mutations we start with. And I'm showing you here just a handful of those that you might recognize. The G27X that sits right around Arizona, Delta F508 over Texas, move on to G542X and G551D, and as we get closer to the East Coast, we hit this guy, W1282X. All right, so some of these mutations are effectively more like potholes. These are the class 2, 3, and 4s. These are the ones that make proteins that can be made usable with our CFTR modulators. So these mutations we can partially or completely repair. So we just might hold off on those while we focus on those others that we don't have any other options for currently, the class 1s. And those are ones that we would be more like a Grand Canyon. They're not a pothole in the road. We can't fill them. We can't get around them any other good way. So, but that's where we're going to start. And I had to shamelessly show my CF backpack, and telling you that, you know, the CF Foundation, we're everywhere, right? We will not stop. But to bring back the analogy, these are all new technologies. And we need to think now about whether or not we have to customize a repair strategy for every one of those 1,700 mutations, or can we come up with something that would work more globally. And there's a new technology emerging that's part of this gene editing technology, would essentially fly over all the road damage, and no matter where it is. And in fact, this type of strategy is being pursued by a number of different groups. And it uses the DNA repair process, but it instead of fixing a particular base pair, it inserts a section or a segment of DNA, and possibly even the entire CFTR gene. And while this technology is still in its infancy, I think it holds immense hope, and I'm going to try to describe this conceptually to you here. So the way it might work is something like this, that instead of repairing a specific mutation, we're going to make a new gene altogether. We're going to insert a piece of DNA, we call it a cDNA, and that's the part of the DNA that contains the parts of the gene that encode the protein and doesn't have the parts that are used by the cell for other purposes. And what that does is it takes that 190,000 base pairs and shrinks it down to about 4,000 base pairs. It makes it a lot more tractable of a problem. Okay? Now, with this one strategy, imagine that there's a place that we'll call the safe zone, upstream of all those mutations. And with this strategy, if we can insert that cDNA, we could have a single method that would effectively fix the CFTR production problem for all patients. Not just those with the stop mutation, but anybody who has a mutation in CFTR. So that's pretty exciting to be able to think about. Okay. Sorry, going the wrong. All right. So in simple terms, what we're talking about here is building an airport before we get to any of the road damage, and we're just going to fly over it. We're going to wave to the Grand Canyon, any other orange barrels we see along the way. That's the idea. Now, whereas all those are very exciting advances, we also, and this has been a CF Foundation strategies, never put all of your eggs in one basket. You never know what's going to work, no matter how good it sounds. And so, as I mentioned earlier, we don't have to just focus on the DNA. We can also think about the RNA as another target. And we would certainly love to have the one-time, one-recipe fix-it-all, but let's talk about some of the other possibilities we have as well. So let's look at the RNA. All right, so conceptually, the simplest thing to do from the RNA is just to put normal non-mutant RNA, CFTR mRNA, into a cell and fix it that way. And there are several companies that are working with the foundation toward this goal. So Shire, Arcturus, and Moderna are all working on this strategy to get RNA into cells. But another approach that's being explored is to exploit a property of the stop codons that are formed, the things that make these X mutations. They all have A's and T's in them. And what some clever folks had recognized was, well, gee whiz, maybe with the T's, we can use a thing called pseudouridylation that tricks the cell into reading the T's in the stop codon as a green light. It's not a stop sign anymore, but actually can work its way right on through there and make some protein. And another one is to use an enzyme called adenosine deaminase that will actually do the same thing for the A's in those stop codons. And again, green light, work on through and make some protein. So I'm showing you one example here of from Josh Rosenthal's lab, a paper he published a few years ago, that in the bottom left you can see cells that were expressing a green fluorescent protein. And what he did in the middle panel was he created one of these stop codons and prevented the green protein from being made. And then he used this adenosine deamination process in the third panel on the right there and showed that you could bring back the production of this fluorescent protein. So he and other labs are using that strategy to see if you can do the same thing with CFTR. Right. And yet another approach is to use specifically modified oligonucleotides. These are short nucleic acid strands that carry the normal CFTR sequence. And they'll, for reasons we still don't understand completely, at least I don't understand, is that then can catalyze correction of the mutant RNA as well. So if we can get those oligonucleotides into the cells, we can correct the RNA and have normal CFTR being made as well. And sorry, that work is being carried out by a company called ProQR. All right, so hopefully you understand now that we're rapidly finding a multitude of ways to manipulate the DNA and the RNA, and all that works great in the lab in a dish. But what about in the body? What do we do there? How do we get it to work and work well? Additionally, which cells do we need to get into? Now, I put up this picture because this was one of the first examples of a vector system to try to get things into the airway cells. And there isn't enough time to go into all the different nucleic acid delivery strategies, but just let me give you a taste of a few of them. Now, the past several decades, there's been an immense amount of work to harness viruses and use their proteins for ways to get DNA and RNA into cells. That's what viruses do. They put DNA and RNA into our cells. We usually don't like that to happen. But one of the first to be considered was adenovirus. And this is just showing an electron micrograph of what those viral particles look like. But the idea here is that it's a Trojan horse strategy, that if a virus knows how to get into a particular cell type, let's use that property, take out the bad parts of the virus and put in the things that we want. And there are many other strategies, liposomes and other things that folks are pursuing to try to get things into the cells. And I'm hoping that we'll hear more about those in the coming months and years. But so with that in mind, we're developing ways to get the editing components into cells, and we have systems coming along to carry out the editing. So our next dilemma is deciding on which cells to target. The cells of the epithelium are clearly places we need CFTR function, but if we target those terminally differentiated cells, the therapy is only going to last as long as the cell lives. On the other hand, if we could get at the progenitor cells, the ones that are the stem cells for the epithelium, correction could conceivably be permanent. And if we can hit those, then we hit that cure that we've been talking about. I'm showing some diagrams that John Engelhardt has provided, and he and his colleagues at the University of Iowa have found that they can actually get into both cell types. So on the left, you're seeing the delivery into the surface epithelial cells, the bronchioles, in sort of the magenta pink color there. That's using a virus, a feline immunodeficiency virus, where he was able to get into those airway epithelial cells. And on the right, using mouse cells, is showing getting into the stem cells. These blue cells that are shown there are clonal derivatives. So it shows that you went into one cell and it divided into other daughter cells. So again, demonstrating that we can actually get into a stem cell population. Right. So how do we decide which way to go with that? Well, the stem cells are certainly more attractive, but they're a harder one to get into. So we're going to try both ways and see what works. The airways and the lungs are very complex, and we're still learning more and more about the various regions, the stem cells in each of the regions of the cell of the lungs. And the only way to do this is to try it and empirically decide what works out. Now, no matter which strategies we pursue, we need a way to see if they work in a living organism. In a mammal with CF disease, similarities to humans, with an immune system, with the complex organ architecture and the multitude of cell types that have to be navigated, those all need to be included. And we have more animal models for CF than any other genetic disease out there. And those include pig, rabbit, ferret, and rat models. But since we're in Orlando, we certainly have to give some press to the mouse models that are out there. And I'm going to tell you a little about some special mouse models that we're hoping to be able to use for some of these therapeutic processes. Now, I've got to go back to our challenges. The first one we need to deal with is that inherent difference in our DNA, the very property that differentiates different species. Remember, we're trying to develop methods that will work on human CFTR on its DNA or RNA. And the DNA sequences in any two species are different. So how can we make this work then? In other words, coming back to our barcodes, the human barcode won't work in another species. So how are we going to use these animal models? Well, we took a very straightforward approach, and that is to put the human CFTR gene into the mouse genome. And this is a process we call humanizing. We certainly weren't the first to do this. And this particular example for CFTR was done by Laura Gowinas on a project funded through the CF Foundation. And Laura's now at the University of Missouri working with Lane Clark. And she created this mouse that has the human CFTR gene inserted, but it's on a different chromosome than the native mouse CFTR, so we can easily differentiate between them when we're producing the animals. Well, Lane Clark and Craig Hodges at Case Western Reserve have found that the normal human CFTR gene will compensate in the mouse for when the mouse gene is inactivated. So without the mouse gene, or without the human gene, the CF mouse experiences all kinds of CF phenotypes: impaired growth, intestinal obstruction, airway inflammation, airway mechanics differences, and others. So it allows us to now look at this as a disease model and how to go about trying to fix it. Well, using that same CRISPR-Cas9 technology that we described a moment ago, Ron Conlon and Craig Hodges at Case Western have generated now the Delta F508 mutation, G551D, G542X, and W1282X mutations in the human CFTR gene in the mouse. So once we get the characterization finished on those guys, which will be happening very quickly here, we can now use those for testing our therapeutic approaches that we're trying, whether they be gene correction or other therapeutic strategies, the read-through drugs, things like that. We now have something that will allow us to assess human CFTR. And we're not stopping there. We're putting more and more. We're not going to do all 1,700, but we're putting more different mutations in the queue all the time, and currently we have the 3849 plus 10 kbc to T and N1303K that are on the docket next. All right, so we now have the human barcodes in our mice, and with a little more characterization, we're ready to try the editing strategies and start the process towards fixing CFTR and treating CF. And I hope you'll appreciate now that our toolbox is getting full. It's now time to start our building. And we have the editing technologies moving along. We have the delivery strategies to get the components into the cells. We have the animal models that will allow us to test these and see if the strategies work and continue to improve them. So now, while we recognize it may be a long road ahead, we're using a new technology that very likely in the coming years will be considered archaic, but we can at least start the journey now and have realistic hopes that we're going to reach our goals. And we'll have the journey mapped out. And our mantra is, and always has been, that anything short of a cure is not good enough. And we're not done until we've made it happen for all CF patients. And with that, I'd like to thank you very much for your attention and also remind you to join us at the welcome reception over at the Hyatt. Thank you.